In today's lecture, we are going to discuss about the membrane protein, which is the most important part of the biological membrane. Membrane proteins. Membrane proteins are the proteins that are attached to or associated with the membrane of the cells or the cell organelles. Membrane proteins have various functions like transport. They transport solute across the membrane. They can be channel protein or the carrier protein. Channel proteins provide hydrophilic passageway through the hydrophobic membrane, whereas carrier proteins are based on the alternate conformation model and they carry solute across the membrane. Second function is anchorage. Anchorage means attachment. They help in attachment of the cytoskeletons to the cell. Next is receptor. They are involved in recognizing and mediating the effect of the specific chemical signals that impinge on the surface of the cell. Example, hormones, neurotransmitters, growth promoting substances are the examples of few chemical signals. One of the major important functions of the membrane protein is its enzymatic activity. Enzymes are involved in the metabolic pathway. Example, GAPDH involved in the catabolism of blood glucose. Next, cell recognition. They recognize some molecules, example, MHC proteins and antigens, and help in various cellular processes. Next, intracellular communication, example, gap junctions and plasmodesmata. Gap junctions are present in animals, whereas plasmodesmatas are present in plants. Other functions of the membrane proteins are uptake and secretion of various substances by endocytosis and exocytosis, targeting and sorting of various modified proteins. They also have a structural role in stabilizing and shaping the cell membranes. Example, spectrin, encheiron and RBCs. Spectrin give RBCs its biconvex shape. Main type of the membrane proteins are intrinsic membrane protein, extrinsic membrane proteins and lipid anchored membrane protein. Extrinsic membrane proteins are embedded within the lipid bilayer. They cannot be easily removed from the cell membrane. Whereas intrinsic membrane proteins are attached to the exterior of the lipid bilayer. They are easily separable from the lipid bilayer, able to be removed without harming the bilayer. Lipid-anchored membrane proteins are lipid-bound proteins that are located at the boundaries of the lipid bilayer with covalent bonding. Now, intrinsic membrane proteins. Intrinsic membrane proteins are also called integral membrane protein. They are amphipathic in nature, that is, they contain both hydrophobic and hydrophilic region. Hydrophobic regions are embedded within the lipid bilayer, whereas hydrophilic regions are outside the lipid bilayer. They can further be classified on the base of they can further be classified according to the mode of attachment to the membrane. They can be integral monotropic protein. As we can see, they are attached only to the one side of the membrane. That is, they are embedded in the membrane only one side of the membrane. Next are transmembrane proteins. Transmembrane proteins means they span the lipid bilayer. They can span the lipid bilayer only once. Example, glycophorin in RBC's membrane. They are known as single pass transmembrane protein. If they span the lipid bilayer multiple times, they can be classified as multi-pass transmembrane protein. Example, band 3 protein in erythrocytes. Extrinsic membrane protein, also called as peripheral membrane protein. They lack hydrophobic sequences, therefore they do not penetrate in the lipid layer and they only lie on the surface of the membrane, as we can see with this diagram. They bound to the membrane by the weak electrostatic forces and hydrogen bonding with hydrophilic portions of the integral membrane protein and perhaps with the polar head groups of the membrane lipid. Therefore, they can be easily removed from the plasma membrane. Examples are spectrin and encheiron. Next is lipid anchored protein. The polypeptide chain of these lipid anchored proteins are located on one of the surface of the lipid bilayer embedded within the lipid bilayer. They can be divided into two parts. One, fatty acid anchored membrane protein. Fatty acid anchored membrane protein because they are covalently attached either to the fatty acid or an isoprenyl group. They can be GPI, that is glycosyl phosphatidyl inositol anchored protein because they are attached to the external surface of the plasma membrane by covalent linkage to the GPI. GPI is a glycoprotein. Thank you. Hope you liked the lecture.